Just give me a second. Hopefully, you are host, but unmute yourself. Yes.
Yeah, fine. Now I'm able to unmute myself. Thank you. Thank you for Deepshika for warm introduction. It was very nice that you gave me a very nice introduction, and I welcome every participants for this brainstorming session on the computer-aided drug design. So with this, I don't want to waste the time. I just move into the slides, and we'll talk more about in the slides. And I think this will be a very interactive session rather than being a single person talking on. So if anyone has any questions or queries, you can stop me or you can message me in the chats. So we can we can discussing about that. So I'll just start sharing my screen. Okay, fine. So Deepshika said, I'm Srinivasan. I'm working as a postdoctoral fellow in Department of Pharmacology in Penn State College of Medicine. I'm working under the Professor Nakole Doklian, who's a pioneer in developing discrete molecular dynamics, which is a kind of protein molecular dynamics simulation which uses stepwise potential in identifying the protein aggregation molecules. And I started working on this during my PhD, and then I started working with him now. So that's a small backstory behind it. So today's topic will be on unlocking the potential of computer-aided drug design, bridging theory and practice. So before me moving into the introduction and other slides, I'll just like to share with you how the presentation is going to be. What are my slides going to be? So initially, I'll be talking about the introduction, and then I'll be giving a brief view on what is the drug discovery process, how it is happening, and what are the process behind it. And then I'll be giving an idea what is structure-based drug design, and then I'll give you a small introduction behind ligand-based drug design, how the ligand-based drug design is helpful, and then I'll give about the pharmacophore modeling, what is pharmacophore modeling and how the QSAR studies have been done, and then virtual screening has been carried out. And then I'll give a brief introduction on molecular dynamic simulations. I think most of them might not known about this, so it will be a very good session on molecular dynamic simulations. And finally, I'll be giving some ideas and thoughts about artificial intelligence in computer-aided drug design. That's how my slides have been occupied today. So let's delve into the slides. The introduction, before me giving an introduction on computer-aided drug design, so I just wanted to let you know what is a drug and what is a target. So why do the people are very much interested in the computer-aided drug design? So the computer-aided drug design, we try to design the drugs in order to target to the protein molecules, which could help in aiding to prevent the disease, or it help in inhibiting the growth of the cancer cells, or it helps in inhibiting kind of aggregates, which can lead to neurological disorders or many other disorders. There are a lot of molecules that could break the protein, that could aid in a catalytic activity, that could aid in inhibiting the growth factors and hormonals or GPCR signalings. So these are like humongous amount of drugs and targets have been till now identified in order to use for the human benefits. That's the most common factor that everyone knows. The intriguing part here is the drugs are kind of molecules that act like a key. So targets are a kind of a molecule that acts like a lock. So it's just like a lock and key mechanism where the drug goes and binds to the target and it helps to unlock themselves. So this can be used in different ways, such as it can be used as an inhibition, it can be used as an activator, it can be used in allosteric pathways, it can be used in different mechanism of action. So the mechanism of action depends on what kind of the process you take for the study, and it depends on what kind of the health disorder you took or you wanted to study. So this all depends on your study based scenarios, but this is the important criteria how the drug works. And there is also a different platform in how this drug works. So there are different mechanisms. It's not only the drug and the target which forms a lock and key model. There is also different mechanism called allosteric mechanism, where the drug can bind to a different position and can induce the changes in the target. So this kind of mechanism is allosteric mechanism. And you also have induced fit mechanism, where the drug and target will have a different shape, and once they bind it together, they change the conformation in such a way that it could accommodate to the drug molecule. So these are some different kind of mechanisms that are prevailing and yet to be identified. Who knows, so these are like one of the most important feature of how a drug and target works, and they try to form a complex. So this is a basic view of what is a drug, what is a target, and how it forms a complex. So the next part is the most important and well-known, well-studied, and well-known directional way towards the development of a medicine. So here, let's see what development of a medicine takes, how many years it may take. So it may take minimum like 10 to 15 years to develop a single medicine. So why does it take 10 to 15 years, and it takes like 40 to 50 million dollars in order to identify one single drug, and how the companies are spending on them? So what is the background behind it? So initially, if you see, when you have a disorder, the first thing the people are willing to do is they try to find a target. So which particular mutation, which particular protein is having a mutation or a genetic disorder, which is having some single nucleotide polymorphisms in them, that is leading to the disorder. So that is the first identification. So that is called target identification. And once you identify the target, then you have to find what is the target, validation set, and about the target, you have to find enormous information about the target. So the first thing is we need to identify the target. That itself is a very big important scenario here. So once you identify the target, then you have to find the compounds that can bind to the target, and then it can lead to the lead molecules. So once it goes to the lead molecules, now in the lead molecules, we try to find the screening of billions of molecules to find the lead molecule. So from the lead, we try to change their pharmacophore. We try to find change the small molecule identification in order to find which is having a higher binding affinity, and it is also not changing the chemical feature of the system, and so on and so forth for this. So all these three process, right? Let me use a pointer here. Okay. So all these three processes are very crucial process. So these three processes are done using computational technique, which gives like 70 to 80% of the industry is using the computational technique in order to identify the lead or the candidate molecules. So if they move on to the preclinical development, and if they fail there, then they return back here and they do this stuff again. So this alone costs them like 3 to 4 years of the time. So once they are into the lead or candidate molecule, then they go for preclinical development. So once if it is success in the preclinical development, then they go into animal models. From the animal models, they go into the human models. From human models, they go into a proof of concept to one particular set of human from one particular human peoples from one particular ethnicity. Then they go for different ethnicity, and they try to find how much the drug is giving the side effects, what is the effectiveness of the drug, what is the efficacy of the drug. So they try to find all those features, and once it is then approved, then it goes to the finally approved stage of developing an FDA approval, and then it comes as a medicine. So consider you are eating a paracetamol tablet. The paracetamol tablet alone takes 15 years of development for each and every process to carry out, and it should come out as a market. So this is a very big process. So that's what we are all delving in, trying to work on this design. So our computer-aided drug design is much more useful in the initial phase of this drug discovery, where we are aid in finding the lead to candidate molecule, and we are the drivers of this particular drug development process. So the next question is, what is the challenge with this? So people will be asking, so it's a very easy process, right? You have a target, you have a compound, you just try to find the fit. So what is the process? What is the so hardest task in this? So here, the biggest challenge is, consider you have a needle in a haystack. Okay, you don't know the size, you don't know the angle, you don't know how the needle looks, and you just try to find only the needle in a haystack. So it will be really hard for you to find the haystack, like a needle in the haystack, without having any kind of external equipments, just to do it manually. So those kind of situation happens when we are trying to manually load or manually try to find the fit and all those kind of things. So that's where computational resource comes into the play, where we try to use computer aided informations in order to find those conformations, or in order to find those information and how a molecule, whether it will fit to the target or whether it will not fit to the target. So this way we try to reduce the amount of the money that has been invested in the development process. So it's a most important thing is the cost cutting. We try to cut the cost in a bigger range as compared to that of the experimental approaches. And then, so the first thing is, like, you have a chemical space of like billions of compounds are being there, and these billions of compounds have been registered in PubChem, ChemBell, or there are so many common amounts of databases which are holding large number of compounds for natural molecules, synthetic molecules, or organic molecules. So these kind of molecules have to be screened in order to get a drug which could able to bind to a target. And the next part is targets. So if you see the PDB database, which we mostly rely on, and currently now we are relying on NMR, cryo-EM technology, and when the AI kicked in, so now AlphaFold is able to give us very high fidelity of structure, which is like 90% relevant to that of the X-ray structure. And so that's the case. What we are now in a situation where we could able to give a sequence and we could able to get a structure. That structure is like 90% similar to that of the molecule that has been crystallized using the X-ray crystal structure. So currently the PDB holds like 2 lakhs amount of PDBs, 1 lakh to 2 lakh amount of PDBs. And these PDBs are the mostly registered X-ray crystallographic, and if you screen them out to the lowest resolution and the best structure, you may get around like 1 lakh structures. So this is the target that we have currently. And now we have to use those 1 lakh structures, or you have to find the target from that particular 1 lakh structures, and then you have to use this billions compound, and then you have to find the combination between these compounds to the validated target. So that's a very intruding, and it's a time consuming, and it is a very tedious process that we try to find out this. And without having any background knowledge on how the compound will be, or what the compound will be, what is the feature of this compounds, or without knowing which particular target or the protein molecule it is able to bind with, and how it looks or how it works, it's a very fine.
Please mute Amna Shehzad. You can do this, sir.
Okay, fine. So that's important thing that we are trying to work on this situation. So the overall work of computer-aided drug design relies on this phenomena. The image what I'm showing here is, so initially this is called structure-based virtual screening. Then it is ligand-based virtual screening. This is called pharmacophore-based virtual screening, where you take the protein ID from the structure and you do the screening. Then you take the 2D structure or the SMILES data set, from the small molecules, and then you convert into the three-dimensional structure. From there, you try to find the pharmacophore features. I'll let you know about what is pharmacofeatures and all those things in the later slides. And then you use computer-aided algorithms. So in the computer-aided algorithms, what we do is we try to fetch all those informations and feed it into the computer process, and then we let no computer to decide how the conformation will be using the biophysical approaches. So we tend to believe on the biophysical approaches, and we tend to inform these systems into the biophysical features, and then we use those into the system-aided drug design in order to compute how the conformation works. So we have, after once we perform the molecular docking, we are done with this molecular docking. I'm sorry, just a minute. Sorry, we're just getting calls. Sorry. So then we could able to work on this MD simulations, and I'll let you know what is MD simulation. MD simulation is molecular dynamic simulation. So that's where we try to find out how this drug molecule works with the protein, whether it is able to bind with it or whether it is not able to bind with it, how far it is able to bind with it, whether it is changing the conformation or whether it is not changing the conformation. So we tend to find all those informations from this MD simulation. And then we move towards the experimental study, and this in the experimental study we try to find the binding affinity or KDA values, and then we try to find whether it is able to change the conformation or not, and we then move towards the clinical trials. So this is a structural way of representing how we are able to find the structure-based drug design in this. So like I said, in the structure-based drug design, the most important feature is the protein. Like you all know, protein is a building block of amino acids. It's a building block of life, and these proteins are being made up with the amino acid. There are 20 different amino acids, and there are other unnatural amino acids which are also involved in this protein participation. And this protein has a differential structure format where they have primary structure, secondary structure, and tertiary structure, the quaternary structure, which try to form the folded conformation, and this folded conformation will lead to the biological process. If there is any small change in the mutation or the synonymous polymorphism or non-synonymous polymorphism in the protein amino acid, it may change its conformation and it may go into the unfolded state, or it may take a multimeric folded state. So initially the protein has two different states, either it is unfolded state or it is a folded state. So there is another important feature that I would like to say here is there are different kind of protein structures where they don't follow this principle of unfolded to folded. It's a two-state process. Okay. So unfolded to folded is a two-state process where the proteins, most proteins are being crystallized and they are being solved using using the NMR, X-ray and NMR technology. But there are one particular set of proteins that are called as intrinsically disordered proteins. So they are a kind of proteins which fall away from this particular shadow. So they are the proteins where they tend to have an unfolded structure, but when they are trying to get a folded structure, they take multiple conformations of the folded structure. So it's really hard for us to map from the folded to unfolded structure because they have a lot of intermediates. So these intermediates will have most effect on the protein, so that we couldn't able to find a very proper folded conformation. That's why they are called as an intrinsically disordered protein. And these are one set of proteins which are like very hard to find a drug molecule because this protein doesn't have a very strong folded structure. So finding a drug molecule which can bind to this binding pocket of their folded nature is really hard for us to find. So that is one of the major milestone that we are facing on this IDPs. And I was working on this IDPs on related to the Parkinson disorder, where alpha-synuclein is one such protein where it has 140 amino acid in nature, and these 140 amino acid are totally intrinsically disordered in nature. So they may take either extended beta sheet, or they may take alpha helix state. They may also be in a totally unfolded state. So it's really hard in order to find them. So most of the people who target those kind of neurological disorders always tend to use the structures which are in an ordered form. So once the people are, when they are found with the neurological disorders, they have aggregates. Aggregates are nothing but proteins that are clustered together. So these kind of clustered molecule together, then they are very strong, or they become like a fibril, and these fibrils are like toxic in nature. So we then, once they are in toxic in nature, then it becomes like a plaques which are formed in the inside the brain, and that's how they are defining how the aggregate formation and leading to Alzheimer's or Parkinson disorder. So the main aim of the computer-aided drug design towards this kind of neurological disorders is targeting those aggregate molecules and breaking those aggregate molecules, which could aid in longevity of their patients. That's a important feature. So that's a part that I would like to share with you all from my experience. So let's come back into this protein structure. So once you have a defined protein structure, two-stated model, like I said, from unfolded to folded structure. So this is a folded structure of the protein, where you can see a lot of alpha helix in the beta sheet structures. So with this protein model, which is obtained from the X-ray, then we take the binding site prediction. So what is binding site? So binding site is a particular cavity inside a protein. Okay, the protein has a large number of cavities when you take in a surface outside. But if you have one particular site where, if a molecule or if you change one particular amino acid, the entire protein's biological function may change. So it is kind of called as a catalytic site or a binding site. It depends on the protein what you're taking with. So we try to find those binding site, and these binding sites are either provided within the literature, or you can use computer aided tools in order to find this binding site. And then you can try to find out which particular binding site has the cavity which is having the ability to change its conformation or not. So that we can find it out. If you have a proper literature on this, then it is very easy for us to find those binding site. If not, we can use computer algorithms in order to find those binding site. That will be a big uh, challenge for us. And it depends on what type of a protein or what type of a molecule or what type of a disorder you take to use, and then you can find this particular binding sites. So once you find the binding site of the protein, then you can move towards the compound library identification. So in the compound library identification, like I said previously, we have humongous amount of libraries. It depends on, like, again, I can say this, it depends on what the user needs. Okay, it's not just like you can use billions of compound to screen. So it depends whether you need to use a natural molecule, or you need to use a synthetic molecule, you need to use an organic molecule, or you need to use the molecules that are different from the plants, or that is from different species. You can use all this informations. You can take a mass spec and you can get all those informations, and then from there you can try to predict the structures. So some labs, they try to get the plants, and they try to sequence they try to use the mass spec, they try to find which is the most highest affinity of the compounds in that particular plant, and then they try to figure it out, and then they try to get it into the smiles format, and from the smiles format you can convert them into the 2D stuff format, and from 2D to 3D you can convert it, and then you can perform the simulations or molecular docking. So this compound library and totally depends on what kind of a study you are going to use. So that's what the most important factor of the compound library. And once you have the compound library, you can do the molecular docking, and then you can do the scoring and ranking. So in the coming slides, I will be talking about the scoring and ranking, what is called a scoring and how they rank between them. So so from this, I could conclusively say that for a structure-based drug design, the most important feature is the target. The most important feature is the target. So what is a target? Like, target may be derived from the experimental or NMR technique. And in computationally also, we can obtain the target using homology modeling on from MD simulations. We can use it. And then we have to find the druggability of the target. That is the most important factor. And then the choice of the binding site. Like I said, it depends and varies between different proteins, and we can use that. And then finally, we have to select the most relevant protein structure. And then we have to find the flexibility, and then we have to find the protonation state of the protein, whether the histidine aspartic acid, how they are protonated, and what is the protonation state. Then we can calculate the pKa values in between them. So all this can be done with the use of the computational tools that are available. So you have a humongous amount of tools that are available, so you can just use them. So it's not a big deal in this, but I just wanted you all to know that structure-based drug design is depends on what kind of a study you are using, what kind of the protein you are using. So once you get to know what kind of protein you are using, I suggest that you can make a very good literature review on that particular protein in order to know how the protein is, what is the amino acid that is architecturally building the protein. So once you get to know about what is the amino acid content and how they are forming the protein, and what are the key amino acids that are important for the protein, then you can like totally modify your protocol according to that particular protein. So that's what I just wanted to convey my information here. So once that information and that particular sequencing of target, if you have found out, then you can go for the compound based structure. In the compound, you can take the entire set of the compounds from the PubChem, and you can do the molecular docking, or you can use it with the pre-processed libraries that are already there, where you can find the stereochemistry, enantiomers, and everything, protonation states, and you can perform the molecular docking algorithm. So, sorry, in the target-based design, the most important feature is the target. You have to identify it, and you have to find the particular binding site, and then you have to find the most relevant structure. You have to find the flexibility of those structure, and then you have to find the protonation state. Then you can go for molecular docking. So this here image here gives a rough idea for you on how molecular docking works on the structure-based drug design. So structure-based drug design doesn't work only on the small molecules, it also works on the peptides. So this slide I have purposefully made here to show you how the peptide works. Since I have also worked on the peptide docking with the protein, so I just wanted to share my knowledge with you all. So like a small molecule, the peptide also goes through the same procedure where the molecular docking is the same, and then we would try to find the conformations. But there is a unique feature with the peptide is like you have a peptide, forming a conformation of the peptide itself is a very tedious process. You have different computational tools to get the conformation of the peptide, but a peptide may be alpha helical in nature, or it may be unfolded in nature, or it may be a beta sheet in nature. So that is how the secondary structural feature of the peptide will be. So once you get to know the secondary structural feature of the peptide, you can start building the peptide. Okay. So once you start building the peptide, next you have to generate the conformation of the peptide. It's just like you have a fragment of a protein. Okay, then you try to take that particular fragment, and then you try to build that fragment, and then you try to find the conformations of that particular fragments. So once you have all those fragments in the nature, then you take the receptor molecule, you find the binding site, and use all those conformation of that particular peptide. So the most important feature is like it's not one single conformation that you take for the peptide. You take like hundreds and thousands of conformation of the peptide, and then you dock it or redock it in the same position of the binding site again and again in order to find which particular pose or the conformational angle that the peptide is able to have a good fit with the protein. So this is one of the most tedious, and it's a very time consuming process in order to find the which particular conformation has a very strong binding affinity with the protein. So this is one of the most important feature in the peptide based drug design that we tend to find. So this is where the image where you could see like hundreds and thousands of peptides that have been bind into that one particular position of their binding site, and From there, we try to use the scoring technique in order to find which particular peptide has the best docking score with the protein. So this confirmation can be obtained using Monte Carlo search, and this works on genetic algorithms which tend to evolve using the degree of freedom and the torsional angles. So that's how they tend to develop into the conformations and conformational space. So what is about the scoring functions? In the scoring functions, it is way to aim the people. What do they do tend to do is, once you have multiple number of confirmations, how do you rank them? So ranking them can be done using the values, right? So how do you give the values? So for giving these values, we need to have certain amount of information that can be taken into the consideration, and it could help in getting us the values. So what are the information that could we take that we could take, which could help us giving the better values and in finding the affinity of the compound? So for that, the initial feature, what the people are mostly using the Autodoc Vina or Autodoc, the people who have developed from Morris Labs in the US, they have initially found that for the molecular docking, you tend to find the energy of the van der Waals, energy of hydrogen bonding, energy of electrostatic interaction, and energy of the internal energy. So all this you combine together, and you tend to find what is the docking energy between the wild type and the ligand in the docked complex, and they'll subtract it, and they'll tend to find how much the ligand has changed its energy or not, whether it is a negative value, whether it is a positive value. They tend to find this. Okay, so here you could able to see this is a ligand which is of a different shape, and this is a receptor. So in the docking algorithm, it takes different conformational search. It can take different positions. Here you could able to see, but you see this, the first one is the very good fit. Okay, so once you try to find a good fit, so visually you can't, if you have a very small number, you can find it. Consider you have billions of molecules, visually finding it will be a very tedious process, right? So for that, we tend to use the scoring function here, and this scoring function will help us to know how much the binding affinity between the receptor and the ligand will tend to form the complex, and it will show the negative the binding affinity will be, the better it is. Mostly we choose that. So that's how we choose from the difference between the wild type and the mutant, and we tend to find which one is having the higher binding with them. The commonly used scoring functions are a force field based scoring function, empirical based scoring function, and the knowledge based scoring function. So the usually people tend to use physics-based scoring functions and the empirical-based scoring function. So what is empirical-based scoring function is where the people usually use this van der Waal, hydrogen bond, electrostatic, and internal. The physics-based, they tend to use the Coulomb's and van der Waal interaction. They tend to find the binding free energy between them. And the knowledge-based potential, they use potential mean force, whereas negative logarithmic of the particular value, they tend to find how much the energy difference is there, and then they tend to calculate the free energy values between them. That's a knowledge based potential. And currently we are using with the AI intelligence, we are trying to use the difference of machine learning. So in the machine learning, they tend to give a cutoff of the wild type, and then they take the difference between them, and they tend to summate the entire value, and then they try to compute the energy between them. So these are the different functions which could give how the scoring function is used to provide the binding affinity of the molecule towards the receptor. So mostly we tend to use with the empirical based scoring function. Different algorithm uses different for functions, like Autodoc Vina uses empirical, or like DFire uses knowledge based potential, and RFDoc RifDoc use machine learning based potential. So there are different kind of algorithm that tend to use different base scoring functions. So this depends totally on the algorithm, how they have developed it. So next we can go into ligand based drug design. So in the ligand based drug design, the most it's a very kind of what do you say, it's just like a very fascinating way in order to find the molecules that are similar together, but they don't have a same properties. And you can able to find the molecules that are like dissimilar together, but they are following the same like having same properties. So the molecules which look similar but doesn't have the same properties, molecules that have same properties doesn't look similar. So these are like very fascinating thing in science, that how the molecules are arranged. Chemistry, it's a very fascinating field, and this is how the molecules are tend to arrange between themselves, and they tend to form different complexes between them. And one particular change of the small molecule, right, these are called as small molecules. One particular change, like here you have NH, and here how the NH has been defined, these can change the affinity between the molecules, right? So how they are oriented, how they are technically oriented will change the affinity, and it will change the property of the molecules. So I don't want to move into the chemistry part now. I just wanted to stick on with this. Let's not digress. So the first thing what people do in the ligand-based drug design is, once they find which ligand has the best binding affinity with the protein, right? They try to find one particular ligand which is working with the protein. So we can't stick to one particular ligand for the entire system, right? So we tend to find different ligands, or we tend to find something which is how do you improve the ligand, right? So we have to do the improvisation in order to find the better affinity, or it can work on better in changing the conformational of the target. So for that, what they do, they first do a similarity search. So this similarity search works on the principle where it will tend to find the backbone architecture of how the ligand molecule is, the ligand molecule that we tend to have is, and it will try to find out, and it will tend to give us all the compounds from the library which is having similar kind of a backbone architecture with small changes. Okay, and then it will give a score. So this score is called as Tanimoto coefficient. This score gives tells us how much these molecules are having similarity. So once we've trained to find the similar molecules, then we check whether these molecules are having similar properties, or they are having a negative properties, or whether they are carcinogenic in nature, whether they are not carcinogenic in nature. We tend to look all those properties next. So this is the first step, trying to find the similar compounds with the similar properties. It's a very big step here, and it is a very fascinating step too. And once we trying to find the compound set, right? Once we try to find a compound set where we have like uh, one particular lead compound, and from that lead compound we tend to obtain like hundreds of compounds which are having similar backbone architecture, and then we use those similar backbone architecture compounds as a data set, and then we perform the molecular docking again. So once we perform molecular docking again with the same binding pocket, we tend to find the molecules which are having better binding affinity, which may or which may not. Okay, that depends on the work. So mostly we tend to get the molecules which are having better binding affinity than our lead compounds. So that's the most important way and most important process where the people are using the ligand based drug design. So what I'm saying is just an overall picture, but if you delve into much more of this ligand based drug design, so you have the features in them. So like I said about the properties, right? These properties are calculated using using molecular descriptors. So in these molecular descriptors are just like they have 3D or 2D features, where they tend to find like hydrogen bond donors, hydrogen bond acceptors, aromaticity, whether they are having a solubility, whether they are having the aromaticity, and all these features. Okay, these are called as features. So these kind of features are being calculated using molecular descriptors, and there are different tools which calculate the molecular descriptors. Dragon is one particular software which has like more than 6,000 features in them, and these are called as descriptors, and these descriptors tend to identify which molecule of the ligand is having a better property towards the lead compound. So that's how people tend to use this. So here the slide will give a very important representation on structure based drug design and ligand based drug design, where in the ligand based drug design you could really see how it is moving. Just a minute. Okay, the ligand-based drug design, you could able to clearly see how the ligand information we are obtaining, and then we are going to pharmacophore modeling, and then we are going to QSAR studies, and then we go to the virtual screening of the compounds, and then we go to lead optimization, and then we go to the drug candidate identification. So the difference between the structure-based and ligand-based drug design is once we have the structure, we tend to get the pharmacophore modeling, we get the ligands, and then we perform the docking in them, and we find the lead compounds, and we perform the molecular dynamics. Whereas in the ligand-based drug design, we have the ligands, then we perform the pharmacophore modeling, and then we perform the quantitative structure-activity relationship for the compounds, and then we perform the screening using molecular docking, and then we perform MD simulations, and we perform the lead. If it has a small error in them, and if it is not a very binding affinity with them, then we go back and we perform again this task from the step 1 to step 2, and again we repeat the process, and then we try to find the drug candidate. So this is how structure-based and ligand-based drug design are different with each other, and that's how what we are going to see in the next slide is pharmacophore modeling. So what is pharmacophore modeling? Pharmacophore modeling is an ensemble of steric and electronic features that is necessary to ensure the optimal supramolecular interactions with the specific biological targets and to trigger its biological response. So there are a lot of words, right, which are like different saying supramolecular interaction, biological target, and all those things. Let me simplify this to you. So you have a ligand molecule, right? So once you have the ligand molecule, you can find what are the features of the ligand molecule. So the features mostly tend to fall in the hydrophobic groups, whether it is aromatic amino acid, whether it is aromatic ring which is present, whether it is sulfur group it is present, whether it is a hydrogen bond donor group is present, or whether hydrogen bond acceptor group is present, whether it is a fluorine molecule, whether it is a chlorine molecule, whether you have a metal ion in them. So these are the features of the compounds. Okay, so these features of the compounds can be extracted using the different softwares. Like I said, you can use these different softwares, you can extract those features. So once you extract those features, right, so you can tend to get the new molecule in such a way that these molecules should have these features. So when we are trying to search, that time we try to search the molecules which are having hydrophobicity, which are having hydrogen bond acceptor, which are having this many aromatic rings in them, like 2 or 3 aromatic rings in them. It should have 2 hydrophobic groups in them. It should have a metal ion in them. It should have a hydrogen bond donor in them. And you can like modify all these parameters, and you can be able to screen the other molecules using this as a base molecule. So once we try to do this, next we try to find out if we change the hydrogen bond donor or acceptor, it could improvise or not. So this can be done using the pharmacophore mapping. So this is what we try to identify whether which particular molecule or which particular site. So you think you have a molecule which is having hydrophobic site, aromatic site, and hydrogen bond donor site. Okay, so you don't know how to change a small compound feature, chemical feature in the compound that could enhance its binding affinity. So you can use the pharmacophore mapping to tell them which particular feature could be an important feature that if you could modify it, it will enhance the binding affinity. So that is the most important feature of this pharmacophore mapping. And there are software and tools which could help us in aiding towards the pharmacophore mapping for this. So once the pharmacophore mapping can be done, then people use the pharmacophore mapping in a machine learning way. Before the machine learning kicked in, there was a way called they can use different properties like where they can find the correlation between the models, and they can build the model. So these are mathematical models. So quantitative structure activity relationship is where they tend to find the active and the inactive compounds, and they try to build a model. So this model is built upon the mathematical equation where once the molecules descriptor values are given, and when you know the target molecule, and it will try to match up with the target molecule, and it will try to show how much the matchup molecule is been relative to that of the target molecule, and it will tend to find out the value to it. So if this value is pretty much closer to it, like a correlation factor, you usually call as a correlation factor. If this value is pretty much close, the correlation factor varies from 0 to 1. And if you have a correlation factor which is above 0.8, then we tend to take these molecules as the molecules which are having some similarity with that of. So that's how the QSAR models are built. And for this QSAR model, they tend to use IC50 Ki value, and they have toxicity, non-toxicity values and properties and all such so on. So like I said, Dragon is one particular software which uses all these chemical descriptors in order to identify what is the important features of these small molecules, and then we could we could aid in building the mathematical model, and we could feed those information into this mathematical model, which could help us in pointing out the correlation factor, and from that particular correlation factor we could select the molecules which are like very much similar to that of the target molecules. So it's a very big process. That's how we tend to find this structure activity relationship using the ligand based drug design. So the next part I would like to talk to you about is virtual screening. So what is virtual screening? So virtual screening is nothing just like a in silico work where you have a million of compound data set. Okay, so you take those million of compound data set and you screen them with one particular target. Okay, it's just like molecular docking, where you take a particular target and you have a binding site and you perform one particular docking for one set of molecule, and you get the target, you get the complex. In virtual screening, what we do is we take the same particular target and the binding pocket and everything. Now what we do is we have a million of compounds, right? So we take these millions of compounds and we systematically provide the docking search for each and every compound, and we store those values in a data set. So all these values are stored in particular data set, and then we could able to print the informations in the system, and from there we could able to find out which particular molecule is being helpful in finding out the highest binding affinity. So that is called as virtual screening. So why this virtual screening is very much important is because it helps in reducing the cost and time, and it helps in reducing the animals in experiment, and it is the most important feature is like we could use this feature of virtual screening in order to eliminate the failures. So experimentally it is so much cost, and if you fail in experimental and then redoing the experimental is like you have to spend millions of money in it. So people are not willing to do that, so they tend to use this virtual screening technique in order to find how much they could able to reduce the cost and time, and how they could able to eliminate the later stage failures in this. So with this, I could conclude on the entire structure based on ligand based drug design to you. So in the ligand based drug design, the target is unknown. Okay, in the structure based drug design, we know the target. We take the three-dimensional structure of that, and then we use the structure based methodology. We perform the protein molecular docking, protein ligand docking. So in the ligand based drug design, when you have a known set of ligand, okay, then you find out the activity of that particular ligand. Then you tend to use similarity search in order to find how much similar compounds you get, or you can use pharmacophore mapping in order to find which particular chemical moiety you can change that could increase the binding affinity with the target. And then you have a QSA or machine learning models where to predict whether the set of the compounds that are being obtained from the library are similarly active or inactive in nature. Yeah, fine. So now I think we are all in the same page with the structure-based drug design, ligand-based drug design, right? So like I said, all the structure-based and ligand-based drug design are fully dependent on static structures. Okay, you take a static structure of protein, and you take a static molecule of ligand, and then you just dock with them using the conformational search algorithm, and then you get out a final complex which is static in nature, and you tend to show that this particular complex is having a better affinity with them. This is how we are talking about all this particular ligand based structure based design. But what happens, okay, consider a small molecule which is having a higher binding affinity with the protein, and you say that okay, this will help in binding, and we can go to the clinical trials. But what happens is So once you do on the clinical trials or when you go to the experimental study, the ligand just gets away from that. So this happens because when the protein is in the dynamic state, okay, the ligand can't accommodate with them, then they tend to move away. In the static state, we could able to find a very good fit, but in the dynamic state, they can't able to maintain that fit. So that's where we tend to use molecular dynamic simulation. So initially, molecular dynamic simulation were developed by Iris Barshall, Michael Levitt, and who got the Nobel Prize in chemistry on developing this technology on molecular dynamic simulations. So this simulation works on the basics of the physics principles, and this is where you could able to see how a unfolded protein, with the help of these force fields and the physics-based information, they tend to form a three-dimensional structure of a protein. And this three-dimensional structure of the protein is the lowest free energy minima in the blue color, where you could able to see it from here. So they tend to form a structure. So I'll show a video on how it is working later. So let me give a small introduction on what is molecular dynamics is. So molecular dynamics is where we tend to assign velocities to the atoms, okay, and then we calculate the force on the atoms. So then we calculate the force using the Newton's second law of motion, where force is equal to mass into acceleration. And then we calculate the force at the velocity, so the next step. Okay, we calculate the velocity at the next time step from t to the t plus delta t. And then then we use these velocities to get the coordinates for the next time step. So this is how we eventually evolve. Okay, this is just like an evolving process. We have a different algorithm like leapfrog algorithm or steepest descent algorithm in order to evolve to different conformation, and we tend to get those three-dimensional structural coordinates from those informations. So then once we tend to do this, we just repeat this process so on to so forth in order to find the lowest free energy state minima of this structure. So that is what we tend to do. And the next important feature in the molecular dynamics is the force field. So what is force field? Force field is set of potentials that we compute or we have pre-computed, we have fed into the algorithm, that how much is the bond angle, how much is the torsion angle for each and every amino acid is. So we tend to calculate the spring potential between the bond angle using the free energy of the bond. Then you tend to use the angle potential using the theta. Then you tend to use the phi angle for the dihedral angles, then the improper dihedral and the proper dihedral angles. Then use the non-bonded interactions like Coulombic interactions and the lambda Lennard-Jones interaction to calculate the van der Waals potentials between them. So you combine all these factors together to find the total energy of the system. So that's how the force field have been defined, and I am not moving much more into this because it will be this is not the topic of our interest in today's talk. So we'll move on to how the molecular dynamics works. So here in this image, so in this video, you could able to see how protein from an unfolded state is able to move into a folded state. This is taken from the NIH lab, and I'm just here showing from the YouTube to you to show how this protein, which is in an unfolded state, you can able to see, which is tend to form a folded conformation as that of the X-ray structure using the molecular dynamics simulation alone. So here you could able to clearly see how molecular dynamics is able to capture the three-dimensional tertiary structure of the protein using the MD simulations alone. Without using any external factors, you can use the MD simulation in order to get the three-dimensional structure of the protein. So this is a fascinating world where we can use molecular dynamics in order to understand how the protein folds and what is that function. It doesn't stop here. The next part is like how the MD simulation is used for the drug discovery. So in terms of the drug discovery, molecular dynamic simulation offers a very powerful tool. Okay, so it helps in mimicking the dynamic movements of the interaction of the atomistic levels. Okay, the movement of the amino acids in the atomistic levels. So here, the most important feature is we tend to find the binding affinity and the ability of the drug which can able to bind to the molecule. So once we are done with the molecular docking, the next part is MD simulation. So here in the MD simulation, we tend to find whether the molecule will have its binding affinity with the target or not. So here it's a very lengthy process. We take microseconds of simulations in order to find how this binding affinity works. So there are different algorithms. We can use shared molecular dynamics. We can use potential mean force to find out it, or we can use a normal MD simulations, or we can use discrete molecular dynamics. So there are like different resources and different works, different MD simulation engines we can use. The most commonly used engine is GROMACS. So it was developed in Germany in the Groningen Institute, and then there is another one called Amber, which was developed by the peoples in US, and then NAMD is also the another simulation engine which has been used for wide long, and Schrodinger has also Desmond to use the simulation engine, and there are also discrete molecular dynamics which was developed in our lab, where we use stepwise potential in order to mimic this molecular dynamic simulation using the Monte Carlo search algorithm. So there are different algorithms and different engines which can be used for this MD simulation. So it finally depends on how which algorithm you would like to use and which one feels better for your study. You can take that. I think from my range of knowledge, I could suggest that Amber is one which works pretty good for DNA, RNA, and carbohydrate moiety, whereas GROMACS works pretty better for the small molecule docking protein complexes. So that's where we could use these informations, and DMD works better for aggregating molecules where they tend to find out how the protein gets into the aggregation zone or everything. So we can use these informations, and we can use it for our study, and it, depending on our study, we can use which particular molecular dynamics engine we could use for our drug discovery process. So once we tend to use this, we tend to find the solvent effect, we tend to find the hydrogen bond interaction, we tend to find the solvent accessibility, we tend to find the binding affinity of the ligand between the protein with this molecular dynamic simulation over the dynamics range rather than the static range. So here I could show you an video which is a drug is able to bind to its particular target, and you can able to clearly see how the drug binding has changed the conformation of the protein, giving it space for the drug to bind and interact with them. So I think it's a very cool video. You could see it for a minute or two, then we could talk about it. Oh, that was a very nice video, right? So here you could able to see from this video, right, how a small molecule upon binding, it could go and fit into this binding pocket where the protein is making its flexibility in order to accommodate this particular ligand molecule to fit into that particular binding pocket. So this is how this fascinating and uh, brainstorming how molecular dynamics works. So it's a world which I think is most explored, and it has been still explored in order to find out how the drug and molecule interacts with the targets and how they are finding their own binding pocket, and they are trying to fit into the molecule. It's a fascinating journey for this. So with this, I'll just stop with the molecular dynamic simulations, and then I'll move on to the AI. So, what is AI? AI is just like artificial intelligence. What people are now, it has become a buzzword where people have been totally using artificial intelligence from ranging from any kind of driving vehicles to healthcare to scientific facts, or you're using in material chemistry or supramolecular chemistry. You take a field, AI is everywhere. So how the AI has been starting to find its way in scientific intelligence is where during the COVID time, where we AlphaFold, Google's DeepMind company, tend to find the three-dimensional structure of a protein. So what they did is they take the sequence information of the proteins from the, and then they try to use the evolutionary informations, and then they use those and they try to train the algorithm in such a way that it could able to predict the three-dimensional structure. So people were initially doing it before they are using it like previously with different algorithms, but when AlphaFold did this, they were able to map it to a very high percentage with the experimental structure. So in the X-ray structure, they could able to map it to 90%, which is one of the breakthrough in the science. And the article was published in Nature, which is like one of the breakthrough of science. There have been so much amount of works that have been done with AlphaFold, and the people are still now evolving AlphaFold for protein dimers, protein molecule interactions. So it all started from there. And the image what you are seeing here is from the David Baker's lab. He's one of the pioneer in developing the protein molecules using AI. So this is called RF diffusion method. So here, so let me tell what is diffusion method. So diffusion method is like you have a fully noise image, okay? You couldn't see any factor what is inside the image. So once you use the AI algorithm, it will denoise it and it will give you a proper image. Okay. The hidden features, what is behind the image, you could denoise it and you can get it through the diffusion model. So what Baker's lab did is they tend to use this diffusion model in proteins, and they try to solve how a protein structure will evolve from the totally unknown structure. Okay. Without giving any information, it tend to form a new protein structures, and they have experimentally validated some of the structures which are giving high percentage of identity with them in the cryo-EM models. It was one of the nicest thing that we have ever seen in the AI, where they could able to get new molecules, and with this new molecules they were also able to design new drugs. The protein-protein interaction also forms the drugs with them, and they were able to design new protein molecules which could able to bind as a drug to the target. So this is one of the most important feature that has been prevailing, and it gave way to the futuristic development of AI in the medicinal field. So traditionally, whatever we talked about, the computer-aided drug design, on structure-based drug design, ligand-based drug design, molecular dynamics, all this are very important feature in the drug designing process. But these are very time-consuming, and we have to spend money on them, and it is not that fast, okay, as compared to that of the AI model. So we have a limited computational efficiency, but in the AI, the computational efficiency has gone in a vast way that they tend to learn at a very faster rate as compared to that of the normal physics-based evolving methods. And this has accelerated the drug discovery process in such a way that people see AI as an kind of a different factor. But in the science field, I think AI is a factor where we could see it as an helper, which could help us moving fast forwarding the situations towards better drug discovery identification and molecule identification, which are not yet known. So this could help in finding a very much precise molecules. It can be used in personalized medicine, or it can be used in formulations. So you name it, the AI has it. So it's just most important field in the world, which is developing so fast. And I think this is where the future relies on in developing of computer-aided drug design. So in this AI, what people tend to use is they tend to have the information from the experimental or NMR or molecular dynamic simulations. Okay, they take those informations and they feed those information into a model. So what is the model? The model is nothing but they are just converting the factors into a numerical values. So consider PyTorch. PyTorch is one of the leading deep neural network model. What people tend to do is they convert the features of the protein three-dimensional structure into a tensor. The tensor is a feature where where the PyTorch takes the information. They convert it into a tensor. Once they convert into a tensor, they use different models like graph neural network or convolution neural network, or they tend to use graph attention network, light attention network. There are different networks. Okay, how you can use this. So depending on what the protein or what is the target of AI interest, you tend to use different kind of network, and from this particular network you tend to form different models. Okay, once you get the model, what you do is you tend to identify how this model works, right? So for that, what we do is we take a data set and we split the data set as training and testing. So we give 75% of the model for training and another 25% as a testing. So we train initially the model with this 75%, and then we take the testing and we just test our model with the testing values. And if you are like very faulty model, then we again redo the model, and then we do the training model until and unless we get a better accuracy on the test rate. We tend to redo this, and there are also some hyperparameters in them which we need to tune it in order to get the model to fit with this. So once we get a high fit model like a correlation factor, we tend to get the correlation factor from 0 to 1, and if we get a correlation factor above 0.9, we tend to say that this is a very best model for that. And once we get the model, then it will be just like you have a virtual screening. If you want to do, you can just do it in a day for millions of compound, which may take a week for us to do in a normal structure-based drug design or ligand-based drug design. But with the AI, we can do it within a day if we have the right resources of the GPU or accelerated system. We can do it with it. That's how the AI has evolved us in finding a better target with this. And there are these different models and different languages are being used. So to be of your knowledge, we could give a small example of how ChatGPT works. So ChatGPT is, or Gemini, which recently launched by the Google, are programming factors of AI where they tend to use the text and they tend to give you the information from them. So these are called as large language models, where they tend to use different parameters. Currently, they have trained it with 7 billion parameters, I think so, for the ChatGPT and Gemini. And then that's why they're able to fetch you a lot of information from them. And one of the most important factor is that they were able to generate images. So Microsoft has this DALI, where they use the diffusion model, like I said, in the Baker's lab. If you give a person is drinking a coffee, it will give you an image of a person drinking a coffee. So just with your words, it could able to generate new images with this. So that's how the natural language processing works, LLM. And then you have different tools which you can be developed with the AI. So our lab has developed UL, Vedusadoc, Home. These are using the AI technique, and currently diffdoc and RIF, RF diff for the AI diffusion method, which are used for this molecular docking algorithm. You can try them and you can let us know how they are working with them, and you can use your works on them. It will be a very nice way that if you can find out different new molecules and using these methodologies, and you'll find it very much interesting with this. So with this, I'll stop my talk on artificial intelligence, and I just want to share with you what are the opportunities. And so these two images, right? The image of a scientist and image of molecule. These two I purposefully generated using the Gemini's algorithm. Okay, it has been developed by Google. I just gave them how opportunities for computer drug design, and it generated those images. So this was not taken from anywhere. So these were like given from a text, and I got it through the AI algorithm. So that's how fascinating it is, right? So that's what I just wanted to share with you all. And so this computer drug design gives a very large platform for us in different fields. So once you have a career, there are like humongous amount of career opportunities in this. So once you can be a computational biologist, the computational biologist for where we develop and apply the computational models to study the drug discovery process, or you can become a drug discovery scientist, or you can just work on X-ray NMR structure. These people are structural biologists. They gave way in developing the data sets for the AI and everything. And you can also be a software engineer. Now the Google, AI, and other companies are much more interested in developing into the science using the AI models, and that has given a very large platform in the computer-aided drug design towards the development of new molecules. And then you have research scientist positions, which could help you in getting to know much more about the disease-related informations, and you can pursue your career as a research scientist or data scientist. Finally, you can also work on this, and if you are much more interested, you can do a startup like how Deepshita started her Omics Labs with her own interest in this. So it's like humongous amount of opportunities are like welcoming with the warm hands on this computer-aided drug design, and that's the end. And I'm just happy that I could share my knowledge and information with you all, and that's why. Thank you everyone.
Thanks a lot, sir. So let's take some questions from audience. Everybody, you can unmute yourself and ask your questions. So there were some questions in chat. Let's start with them. People were asking regarding corona vaccine and how this vaccine just came up in the market within just few months. Let's start with that question.
Okay, I couldn't define. Okay, so I'll start with, does it require special computer skills for drug discovery? No, you don't require special computer skills. You just need all your interest in order to learn for this, and everything starts from there. So you don't require any special skill set for this, and it's not a purposeful need for this. Anyone can start doing this, so it's not a basic background of this. Okay, then you must know drug discovery. So we have Autodoc, Pyrex, Pymol, and other computational biology. Those doesn't require skills.
Just okay, let me.
Antizenet, where is your slippers? Go and wear your slippers. Don't.
I request those who have some questions, please unmute yourself. Otherwise, please don't unmute yourself.
Hello, sir.
Mir Muhammad is here. Please ask your question, Mir.
Yes, sir. Hello, how are you?
Yeah, I'm good. Mohammed, yeah.
I would like to ask a question that how computer-aided drug design is applicable in designing and formulating cancer studies with more reliable and efficient results.
So it all depends on how you work on this tensor studies. Tensor studies is like you are using a deep neural network in order to find the models, right? That's what you're trying to ask, Mohammed, right? So Mohammed is muted.
Mir Muhammad, please unmute yourself.
Yes, got it. Thank you.
Yeah.
Hello, sir. I have a
Thank you
question.
so much.
Yeah.
Yeah. First of all, thank you for delivering this absolute good lecture.
Yeah. Thank you so much.
Sir, my question is, I'm a chemistry undergraduate, sir, and I have an interest in inorganic chemistry and that too mainly in medicinal side.
Okay.
So, sir, yeah. So, will this computer-aided drug design be beneficial for me? as an future inorganic medical site?
Yeah, see, the world of AI and computer drug design is used in every field, right? So it depends on what is the final position that you are going to choose, whether you are going to be in healthcare professional or whether you are trying to find a target. Okay, it will help you in finding the molecules that are similar to that, or whether if you are if you are like organic chemist, you try to use mass spec and then you try to find the CAS number of the molecules that you obtain, right? And then you just deposit them in the PubChem molecules, or right?
Yeah.
So
Yes, sir.
those things are very much important in the field, right? So once you get to know about those molecules, it will be easy for you to learn all this techniques. So these are techniques which can help you go through this. So
Right.
it's not that you don't have to know anything. Like you can use this. It depends on your idea, or how you would like to pursue your career further.
Yeah, basically my interest will be is in Parkinson, Alzheimer, like neurodegenerative diseases and the
Yeah.
cure.
Yeah, then
Yes.
you can pretty much you get a better good knowledge on this computer aided drug design, because I worked in this field. I think this is a very good field where you can start working on neurological disorders and everything. That's a very
Thank
good.
you so much for answering, sir. Thank you.
Sir, people are asking regarding free softwares for CAD studies.
So free softwares for computer-aided drug design, there are like humongous amount of free softwares. Like Autodoc is a free software, Autodoc Vina is a free software. Now there are like after the AI has kicked in, you have a lot of web servers which are very easy for you to run it. Okay, so you just have to search it in the Google and you get like diffdoc is free, and the docking algorithm Medisodoc, what we have developed in our lab, is free, and Ohm server, which is used to find the allosteric site, is also free. Like you can do the molecular dynamics, the softwares for using molecular dynamics also free. So there are some paid softwares, like Schrodinger has some paid softwares, and Discovery Studio has paid software. Other than that, most of the softwares are free to use.
Okay. So Ghazi is asking regarding roadmap to follow where we can master this field, because probably Ghazi is a biologist and he want to specialize in pharmacology.
So if it comes to a roadmap, we can't be very assured of what their target will be, because the field of pharmacology is a very big field, and you can just say that I'm working only on this computer-aided drug design. So whatever we talk today is about computer-aided drug design. If you have wanted to pursue a field in computer-aided drug design, then you can learn some of the tools. How could you use these tools, and or how could you develop these tools in order towards healthcare? You can know much more about the protein ligands and the compounds identification. So that works much more better in that way. But if you are taking experimental pathway for doing towards science, moving towards the science, then you can take the pharmacology. You can use different techniques, algorithms, in vitro, in vivo studies, and you can use this. So it all depends on what is the goal that you try to achieve, in which particular way that you try to move forward the science with.
Siddharth Mishra is asking which language is important in structural biology and computational biology.
So if you ask about the programming, I would say like Python is the base course for this. So once you know about Python, you can learn other things very easily. So Python is the basic need, I think so, for this.
Pushkar was asking how can one combine MD simulation with AI, but probably you have answered this question already during your session.
Yeah.
Hello, sir.
Yeah.
Hi, sir. Omkar Sonavne from Pune. So I just wanted to
Yeah.
ask you the question from the regulatory perspective. So suppose we discover a drug, as you said, through all these processes. So what should we go for if we want to patent it? So do we need to patent a process or a drug?
No, mostly people patent the drug. Okay, they don't patent the process, and they do the patent the process also in some cases, but mostly people used to patent the drugs.
Sure. Thank you.
Hello, sir.
Yeah.
So this is Sahana, and I'm from VIT. So
Yes.
my question is, we in molecular docking, when we are finding out, suppose say a binding affinity, we are getting scores and hits, and we get RF value and all those. So what is the significance of that value? Like, what does that value mean? Why are we selecting that for the next step of molecular design? So the scores, rank, and hits. So what do they signify?
So which program are you using on for this?
I used Pyrex.
Okay, Pyrex works behind the Autodoc, right?
Yeah, correct.
Yeah. So it's just Autodoc. Pyrex is just a visualizer for Autodoc. Okay. Autodoc Vina, they use Pyrex. So here, if you getting a score where the values are like you say the negative binding energy between the protein or something like that, which shows us like how much far the drug can bind to the target. Okay, these values have their own definitions. So if you go to the Autodoc Vina website or Autodoc website, so initially people tend to use the binding affinity values, which is always in the negative value, which we get a better binding affinity. So that's how we tend to use. But so it also depends on what kind of structure, what kind of compound you take. Okay, just whether with the values, I couldn't give more information or detailed information on this. So because it depends on the project, what kind of a project you're taking, what kind of a molecules you're taking. So it's a very specific thing. I can't give a very general answer on a very specific question.
Okay,
So
sir. Okay, sir.
yeah.
Naina is asking, we are using Autodoc Vienna. Does it give the exact scoring function for plant ligand-based chemical compound docking?
Yeah, it will give you a scoring function for the plant ligand. So any ligand you take in Vina, it will give you the scoring. It uses a scoring.
Sorry, by mistake, I mute you. Somebody was just interrupting the class. Please unmute yourself, sir.
Okay, fine.
Please continue with the answer.
Okay, so Vina gives binding free energy, right? It depends only on the compounds that you are using in the target. It doesn't mean that it's a plant compound or it is from natural source compound, organic compound. It doesn't belong with that. So if you have a compound, you use the Vina, it will give the scores. So those scores will tell you how much the compound is having the interaction with the target. That's it.
I suppose that it answers your question, Naina. So Noor is asking, can you explain how to calculate PMF?
I think that's a very big question. That PMF calculations are done using a equation called for potential mean force calculation. So it's a very big study. I think people who are interested in this, they can read about Andrew Leach's book on molecular dynamics, and they could get to know about how this potential mean force is calculated. So for this, what they use is the lambda factor, and for this lambda factor, what they tend to do is they tend to take each and every step, and they tend to evolve of the protein from their folded situation to unfolded situation, and they tend to find the free energy funnel of how they are trying to form into this particular information. So it's a very big study. I think just giving one small information won't provide you much details. So it's better if you could study using the Andrew Leach book on molecular modeling, which could give you a very good explanation on potential mean force calculations.
Right. Sahana asked, why do we consider protonation state like what it is in relation with drug discovery?
So like I said in my talk, protonation state is one of the important factors where histidines or arginines will have these factors where how they tend to form, okay, how they tend to form the interactions with them. So that is the most important key factor in the chemistry. So without having the knowledge of protonation, just using histidine molecule alone is not a very good way to identify the drug molecules binding with them. So we have to get to know about this.
So does it mean that histidine, arginine, and those amino acids are the ones that bring the van der Waals forces, hydrophobic forces, and all that? Are they the one inducing those forces?
Yeah, they are the one that are important, right? If you have a histidine in your binding pocket, it is very much important to find out whether it is in a protonated state or whether it's in a deprotonated state. Okay, so it's like I said, this all depends on the target. So if you the people who have solved the target, right, using X-ray, they would have mentioned clearly in the binding pocket region whether this histidine is a protonated histidine or it is a non-protonated histidine, and you have to get to know about the in detail and depth of this. So once you get to know about the detail depth of this, then you can use that information and you can protonate them and you can perform your analysis, which will give you a better results and better identification of the drug rather than simply using a histidine molecule.
Yes, sir. Thank you, sir.
Naina was asking how to find the exact binding site of protein using free softwares, because using BioVia Discovery Studio, it is feasible, but what are the freely available software which can be
No,
used?
I better tell them to use literature survey rather than before using the softwares. You can use literature surveys to find out whether it has been experimentally proven if there are any binding pockets available. If there are any experimentally proven binding pockets, I could suggest them to take that particular binding pocket rather than using the computational.
Right. So again, people are asking about the same thing, whether coding skills are integral part and it is very much necessary, or we can go without it, because some other automated softwares available in the market where just you need to provide the command and everything will be taken care by the software itself.
Yeah, you can use it, but I personally prefer that knowing a coding language, that to Python is very easy software that you can understand it very easily, which helps you to boost your learning or your coding knowledge. You don't have to be like a software programmer for using Python. You just can understand how the Python works and you can use it to automate some of your process. Rather than that, without knowing that also you can work, but after some point of the time, I think you might need a programming language which could help you much more better.
Right. Trishita, please unmute yourself and ask your question. She has raised her hand.
Thank you, ma'am. Hello, sir. Good evening.
Yeah.
Am I audible, sir, properly?
Yeah, I can hear you.
Okay, thank you, sir. So my question is actually I was currently investigating on the disease called leishmaniasis. So in there I was actually I was undergoing a comparative study with phytocompound and a known synthetic drug. So in there like the result in case of molecular dynamics, I found that the hydrogen bond analysis in there, the hydrogen bond which is formed with the particular enzyme and with the phytocompound, it was less than the synthetic drug. So the synthetic drug formed a higher hydrogen bond in comparison to the phytocompound. But the other analysis such as the DFT as well as virtual screening with Autodoc Vena, then molecular docking with Autodoc 4.2.0, they all gave the binding free energy is high in case of phytocompound. Only the hydrogen bond formation, it is less in case of the phytocompound and high in case of the synthetic drug. So how to interpret this in favor with like to the phytocompound? What is the best interpretation? Like how can I support that the phytocompound is the best one like that? So another thing I should add actually, that the MMGB assay also it's given the result that the Gibbs free energy, like the bind Gibbs free energy, it is high in case of the phytocompound in comparison to the synthetic drug. So all the results are favored with the phytocompound, not with the synthetic drug. Only the hydrogen bond analysis. So how to interpret that?
So you see, the hydrogen bond is not a only key factor in energy. Okay, the energy factor, in the binding affinity, if you take the energy factor, also comprises of the hydrogen bonding. Okay, even during if you are using MMGBAC, right? So MMGBAC is molecular of Poisson Boltzmann, right? You are using Gromax MMGBAC, right? Hello, are you there?
Yes, sir. Yes, sir. Yes, sir.
Okay.
I just.
So you're using Gromax MMGBAC, right? So
Krishita, you need to unmute yourself to answer the question.
Yes, sir.
okay. So in case if you are using Gromax, then I think that you better stick with the results from the MMGBSA because it's a dynamic process. So even in the during the dynamic process, you could able to see how the binding affinity is still better with the phytocompounds rather than the affinity of these organic synthetic compounds. I strongly suggest that you can show this as a result in order to show how the binding energy of the van der Waal and total energy if you compute from this MMGBC, right? Solvent accessibility, total energy, and everything is much more better in the phytocompound rather than the synthetic compound. So docking, molecular docking is a small factor to identify it, but if you do molecular dynamics, you could get a clear picture of it. I think you already have a clear picture and you are good to go with that.
Yes, sir. I have a clear picture actually. Just you said I undergone the SASA analysis also, and in there also I got better result in case of phyto compound than the synthetic drug itself. But sir, my question you like the hydrogen bond analysis. Although I give some sort of interpretation, and I want your recommendation on that. So I gave an interpretation that although the hydrogen bond, like the formation of hydrogen bond in case of the phyto compound with the particular enzyme, it is low, but still I give the interpretation because I found somewhere in literature that the hydrogen bond analysis it is like it plays pivotal role in case of protein conformation rather than the stability. Is this true, sir?
Yeah, it's an important factor. See, hydrogen bond is an important factor. I'm not telling it's not an important factor, but if you are considering the interaction energy between the molecule and the compound, right? So hydrogen, what I'm suggesting is hydrogen bond alone is not an important factor. So there are different other
Okay.
factors like van der Waals and other factors which is important. So if you just want to stick only with hydrogen bond, then I think that might be a very intriguing factor for you. So you can choose other important features and you can just show them how the features are important. So only hydrogen bond alone doesn't contribute towards the energy, right? So you can check what are the other factors that is contributing toward the energy, whether it will be van der Waals or electrostatic. You can check that and you can give the information.
Thanks a lot for this answer, sir. Hopefully, it clear all the doubt. The next question is, somebody is very curious regarding the COVID drugs that just after the COVID wave came, this remdesivir drug came in the market. So was there any evident input of computer-aided drug designing in that?
Yeah, people have been working vigorously on this computer-aided drug design, but if you are asking about the mRNA structure of the vaccine that they try to use it, we really don't have big insights because those are all patented and they don't give all those information. But I think every industry which is working on this mRNA or vaccines or development of vaccine, they tend to use the computational resources in order to find how it's working, how it is binding with them, how the unlocking happens. So with the initially people use the cryo-EM structure of the COVID, I think so, from the Nature paper. Okay, so I think so that is the one of the most important factor in identifying the mRNA vaccines. I think so. So I'm not much more sure about it.
Okay. So I guess this is it. Anybody who is interested in this question answer round and want to ask further question, please unmute yourself and ask the question. Otherwise, we will close this session. Mukunda asked, how can Monte Carlo simulation be integrated into computer-aided drug designing methodologies to enhance the accuracy and efficiency of predicting molecular interactions and drug binding affinities for potential cancer therapeutics?
So there are different softwares that have been using Monte Carlo algorithm. So you can use that and you could able to find the answers for your particular drug or target of interest, what you wanted to study. So it all depends on how you use which particular model or algorithm for your study. It depends on that.
Thank you, sir. Shruti want to ask that is there any direct path to do the PhD in computational biology after B Pharma?
Yeah, you can take the integrated PhD. If you want to do it in India, you can take integrated PhD programs and you can just directly work on biophysics or computer drug design. So that's a very direct pathway. You can clear the gate, you can just directly appear in the interview and you can work on whichever professor's profile you like, you can work on it.
Mustafa Said asked some question, but I am not pretty much clear about what he is asking about. I'm just iterating the question. He is saying that if in QSCR model, how I can get the IC50 or EC50 of such compounds from databases like Chamber.
So yeah, there are informations of IC50 values in the Campbell itself for each and every molecule. So you just have to fetch those informations. So it's a pretty straightforward answer.
Okay. Akanksha Raut asked that firstly, she is very much thankful for such a insightful talk. And then she's saying that how do we know which kind of ligand library to choose and how can we choose ligand library?
So it all depends on what is their target, what is the compound they wanted to work with. So it's a very, it's a general answer that we could give it. So it's not a very specific thing that I could say with this. So it all depend on their study, how they are working on what they are using.
Okay. Trishita was asking that what is the best interpretation of hydrogen bond analysis after molecular dynamics studies?
So I think I have answered that question.
Yes, probably yes. Is the Linux mandatory for running Autodoc Vienna? Abhishek Pawar.
No, that's not mandatory.
That's pretty much straightforward answer. Okay. Siddharth Mishra was asking what can be used for heart patients, structure-based or ligand-based study drug design?
So again, there are some general questions, I think. So this are like, it depends on your study. So we can't say it out. It depends on how you work.
Very right. This is genuinely a very loose-ended question. So Kriti Singh asked that if I want to choose a career in drug discovery, which topic I have to focus on? Because there are many things. So what is the most important topic on which she should focus more to get better career opportunities?
So it depends on what kind of study you choose with. It is not about you choose your career depending on what kind of study you choose with. Okay, like you want to work with a professor on molecular docking or dynamics, you can work with them and you can work on the computer drug design part and you can choose it, or you can work with them to develop a tool in order to develop for this. You can aid into the computer drug design, or you can use AI method or models to work with that. So it depends on your interest of research or where you can choose with.
Right. Santosh Prasad Chaudhary Kurmi asked, if we prepare protein, but during molecular docking, that prepared PDB missed some residue and have bond break, and using such protein, we get binding energies. That obtained value is reliable, or we need to recorrect that protein? If yes, then how?
So again, this is a very general question. Without knowing the background of what they are doing, just we can give a simple answer behind this, which will have a misleads to this. So it's only on their study, I think. So they could work on with the different parameters and achieving how this works and how they get the binding affinity, and they can work with that. I don't think I can give a straightforward answer to this. because we don't know the background of their study.
Right. I again request everybody, please unmute yourself only if you want to ask some question. Otherwise, it is creating nuisance. Anyways, Meher is asking about density functional theory. Can you please show some light on this?
So I don't understand how could I show light on density functional theory.
So she's asking why we do this after MD simulation.
So you don't have to do DFT after MD simulation. It is not mandatory and it doesn't involve in MD simulation. So it is just a molecule level study. MD simulation is like a protein level study, which is two different case scenarios. I think so.
Right. So Nitish is asking about that which research institute we will choose to pursue PhD from foreign, from the same field. For example, so basically he is asking about that which research institute is available for PhD for Indian students in computer-aided drug designing field.
So every university in US is available. You can just contact the professor and you can clear your GRE to fill exam, and then you can apply here, you can get a fellowship and you can start working on your graduate program. So it all depends on how you contact a professor, and if he's interested with your work or your research proposal, then you might start working with it.
So the basic path is this: you need to clear the GRE. Is this mandatory?
But there are some universities, I think so, they are not taking GRE, but most of the universities take GRE score, GRE and TOEFL score, and they tend to use that in order to take up the students into their induct the students into their programs.
So the simple process is this: you need to go through GRE or TOEFL.
Hello.
Just give me a second. Let I complete the question. Anyways, please ask your question.
Hello. Can you listen me?
Yes, we can listen. Please ask your question.
Yeah, I've got another question. That is, what is the most important factor in the binding of ligand and receptor? Can you listen me?
Yes, we can listen.
Yeah.
Hello.
Yeah, tell me.
Yes, we can listen you. Please ask your question once again.
Yeah.
So probably he is not here. Anyways, so I was asking this thing that the process of getting PhD abroad is pretty much straightforward, that you need to go through TOEFL or GRE, and after that you need to approach the professor of designated university where you want to get admitted, and then based on his response or her response, you will get enrollment in PhD, or there are specific examination also in this process.
No, once you talk with the professor, then there are like graduate funding fellowships. You can apply for the fellowships, and then they'll guide you through the process. So the most important thing is you have to clear the GRE and TOEFL, and then you have to get the approval with the professor. Once you get to know with the professor, then you can start. Then they will help you out in this. So it varies from university to university. So the most important criteria is finding a professor, and if he is interested with you, then you can.
Right. So I think that's it. If anybody has any question, please let us know. Otherwise, we need to close this session because there are only 15 minutes left.
Sir, I have a question.
Yeah.
Please.
Yeah, sir. Pursuing career in medicinal chemistry, shall we go for inorganic chemistry or the organic chemistry?
So it depends what is interesting you much more to drive towards it. If it is organic chemistry or inorganic chemistry, if you find out working on this in a rotation labs for 3 months, then if you are interested much more in which lab, if you can find out, then you can pursue the career there, I think so. Rather than just simply selecting them, you can just work on the lab for 2 to 3 months, and then you just can use that.
Okay, okay, right. Thank you. And also, sir, what's the criteria of working in those labs?
No, you just have to contact the professor and you can just let them know that I'm willing to work on this, but I just don't have enough idea. So I'll just do a rotation in your lab. So which one is perfect? I could just choose over it. So you can just talk to the professor.
Right. Thank you so much, sir.
Saranya Devi is asking about that in so many interviews, she was rejected because the work done was only in silico. So she is asking about ideas how to overcome this.
I really don't can give I don't know how to give an answer to this because I don't know what
think
is the.
so that if she is applying for PhD or probably in her case she's applying for postdoc, so probably she should target such sort of universities and she should apply to such sort of professors who are working in silico only, right?
No, it doesn't have to be it depends on what kind of work they do. It doesn't have to be in silico or like I really don't understand like rejecting interview is a different part and improving in silico is a different part. So in silico there are different fields, AI, machine learning, ML, computer aided drug design. You have genomics, you have proteomics, you have next generation sequencing. There is tremendous amount of room for this. So it depends. Okay, so if there is something need, you just can find out which professor or which lab is working on your area and you can contact them. That might give you a better opportunity rather than applying everywhere.
She has done her PhD from VIT, if I'm not wrong, and she is trying for postdoc, and in that she is getting so many rejections, and that's why she's asking this question. Sakshi, you want to ask any question? Yes, so probably she'll ask this question to you directly through LinkedIn. Probably you are open for this idea.
Yeah, I can just let her know which might the only thing that I can suggest now is you can search the labs which your work fits in with, and then you can choose that particular lab and you can mail to that professors and you can just contact them. I think so that will be a better idea rather than choosing everywhere, because that's how people tend to choose postdocs position. So it's also about your skill set, and if that doesn't fall within their lab, I think they will be not interested in you. So you have to find which lab fits with your skill set and how the lab is will be for you. So you have to choose all those informations.
Right. So I guess that's it. Thanks a lot, doctor. Thanks a lot. It was
Thank you so much. Thank you, everyone.
It was genuinely a very insightful session, and hopefully everybody enjoyed this. And definitely we'll get back to you, and we will provide all the questions in the attendance sheet itself. There is a column that if the attendees want to ask any question to you, they will put it there, and we will forward that to you. Please answer those questions. So hopefully everybody knows Dr. Omics, and if there is somebody who don't know us, let me explain who we are. So we are Dr. Omics Labs Private Limited. We provide molecular diagnostic services. Along with that, we also provide bioinformatics-based trainings and courses. There are basically two arms of Dr. Omics. The first one is Molecular Diagnostic Lab, and the second one is Dr. Omics Research Lab. So we are in strategic partnership with IIT Delhi, as we are in process of incubation at there. And apart from this, we are MSME certified, Startup India certified, ISO certified, and now the most important part is this: we are Skill India certified. So few of our courses are Government of India certified, right? So recently we got AWS grant for research. We have successfully trained more than 5,000 students, and we are pretty much popular on LinkedIn. So if anybody is interested in monthly webinar or they are interested in services provided by us, I request everybody to follow us on LinkedIn. There we are pretty much active and you will get most of the information regarding us. Apart from this, recently we have signed 5-year-long MOU with Andhra Pradesh government, and we are providing bioinformatics-based trainings and courses to Andhra people, Andhra students. So talking about molecular diagnostic lab, recently we have developed a very beautiful test regarding hair genomics. Previously, we were providing cardiogenomics, oncogenomics, diabetomics, gene resistance, and check my health. But now we are coming up with hair health test. This is a saliva based test. You just need to provide a saliva. We will extract the DNA, and using that, we will provide you with the insight into your hair health. So if anybody is interested in this, just give me a second. I need to. This is quite unacceptable that being even after being educated, you people are not able to understand a simple fact that if you want to ask anything, please unmute yourself. Otherwise, please don't unmute yourself. I'm pretty much disappointed about this. Anyways. Archana, please mute yourself. Otherwise, I have to remove you. Anyways, let us start again. We just want to make this session more interactive. That's why we are providing you with the facility to unmute yourself, but somehow, anyways. So regarding vertical 2, we are providing 5 type of courses: next generation based, computer aided drug designing, AI ML, advanced R programming, and Python. Every month, we come up with a standard 6-month long-term industrial bioinformatics internship. Tomorrow, we will be introducing you people with industrial bioinformatics-based 6-month course focusing computer-aided drug designing. If anybody belonging to this particular webinar is interested in this 6-month course, please let us know by typing yes in your attendance sheet. We have provided such type of option there. If you people are interested, please say yes there. We will provide you 2 free classes so that you can get to know that what is the process and how we are going to teach, what would the quality. And all of you must be knowing about the course features, that it will be highly professional course, and we will provide you with exposure of industry practices. And apart from this, the first 5 students of every batch will be helped in publishing their research project as a research paper. We will help in development of project for each and every student, but based on the attendance and based on the weekly test, we will select first 5 students and we will help them in publishing their project as a research paper. It will be a very comprehensive course, and in this we will teach you from basics to advanced level, and we'll start from how to fetch data from different type of databases, and along with that we will teach you Python and R. And after that, we will teach you a very specific course of computer aided drug designing. This is my team, and thanks to all. Somebody has raised hand. No. Okay. Abdullah Mehboob, please unmute yourself.
Yeah, thank you, ma'am, for this. And ma'am, I want to ask, ma'am, what's the curriculum and sorry, what's the fees of this course?
You are a foreigner student, right? You belong to Pakistan, I guess? Abdullah. Okay, so for Indian students, it is 35,000 plus GST. That will round up around 41,300 something. And for foreigners, it is $450 all inclusive. So for webinar students, there will be 10% off. This is it. I guess this is the complete thing that I would like to convey. And if you people genuinely enjoy the webinar, please rate us on Google and let us know how we can improve ourselves. And if you people are interested in hair genomics test, please let us know. We will provide you further information about it. Thanks a lot, everybody, Dr. Srinivasan, for giving this beautiful session. And thank you, everybody, for being so patient. Thanks for being so interactive. And yes, I would like to complain here that somebody was unmuting themselves without any purpose, but let it be, it is part of the game. Anyways, thanks a lot. Thanks a lot. I guess that this is end of this session and we are just signing off. Thanks a lot.
Thank you, ma'am.
I would like to mention one more thing once again. Please don't forget to mark your attendance on the attendance sheet. Otherwise, the certificate will not be provided, no matter what. Because you people will send us mail again and again that I was there in the webinar, I was not able to just mark my attendance. Attendance sheet is open till midnight. Please try to mark your attendance. That's all. Thank you. Thank you very much. When it will be provided within a day or two, because there are so many participants, we will take some time to just design this thing. Okay then, thank you.